Welcome. Thank you all so much for coming online and in person. This is a really memorable, kind of emotional event in so many different ways. So thank you so much. I want to start by mentioning some of the people who have put a lot of work into this, shown at the Mariners game recently and a scientific conference. The folks who have contributed are listed here, and the kind of the heavy hitters that really put many hours, not just a few hours in, are in blue here. So thank you all for contributing. Our just going to give you one slide on why we're here. I think everybody kind of knows about Merkel cell carcinoma, but about, hi mom. Yeah, just go in the middle. That's fine. Thanks, Samira. Okay. Yeah. Thanks, mom. So about 3,000 new diagnoses of this cancer per year in the United States, going up faster than almost any other cancer because of this weird relationship with age. We don't fully understand it, but we think the immune system plays a big role in keeping the cancer from coming. And as we as a country are getting older, we're a little bit less protected from this cancer. It comes back more often than most cancers. About 40% of all patients will have it recur in some way or another. And these are the risk factors: advancing age, sun exposure, fair skin, immune suppression. And we've all had exposure to the Merkel virus, which we'll talk about just a tiny bit tonight. It causes about 80% of the cases of Merkel cell carcinoma in the United States, whereas sun or ultraviolet light causes just 20%. We're reasonably good at doing the surgery and radiation needed to control this cancer, to get rid of all that we can detect of it. But after that is when the 40% comes back. And that's very tricky. And some patients have higher stage and have a higher than 40%, and some are lower with less than that. That's been kind of just getting fine-tuned over the past years. But the big change was immunotherapy with 2 FDA approvals. I'll say a tiny bit more about that in a few minutes, with Bavencio and Keytruda being names that many of you know. The frustrating thing about that is less than half of the people with advanced disease who get one of these drugs will persistently benefit from it over time. So that's the biggest single challenge we face with this cancer, and we're going to tell you tonight about several exciting developments in that area. So there's huge progress, in other words, but there's still a lot of things to be done. Here are 4 quick, really glorious highlights. I want to highlight Thomas Pulliam, a wonderful MD PhD student who is in his last year of his graduate studies now. And after distilling down billions of pieces of evidence from dozens of patients, he's come to a very interesting, maybe not incredibly surprising, but the first time it's ever been clearly seen, we think, conclusion that if you don't have these guys, Merkel-specific killer T cells, in your blood, it doesn't really matter what drug you give or whatnot, you won't respond. In contrast, if you've got a good amount in your blood, you're very likely to have a wonderful response when the immune system is stimulated. And this has super interesting implications. It means that if we can increase the number of those killer T cells in the blood, and we've got like 4 or 5 or 10 ideas about how to do that, maybe we can cure more patients. And the really cool thing about that is we probably don't even have to make them sick either, because if we give them good T cells that target the cancer, doesn't make people sick. The other drugs that just turn on every darn bit of the immune system can make some people sort of sick, but the T cells themselves, we give them the right ones, kind of work magic without side effects. So that it's just that it's hard to do that. So that's super exciting. This is one of those ideas to generate that immune response. A new $4 million grant given to us and our collaborators in Denmark as a completely new way to generate more of these guys that will kill the cancer. And based on that money, we do have a grant coming to Seattle here in a year or two as that technology is developed. I mentioned a moment ago that we had our first patient treated with a therapeutic vaccine a few weeks ago. The concept there is keep the immune system strong against the virus and stop the cancer from coming back. Because as soon as you chop out the virus, the immune system falls down in its response very quickly. So we want to change that and see if that helps to keep the cancer away. And then we talked last year about an ATR inhibitor. The crux of that is we think it can kill cancer cells in a way that will make them visible to the immune system. So that could really work together with these other drugs and not have the same kinds of toxicities that just bludgeoning the immune system and saying fight everything tends to have when it destroys our thyroids and our guts and our lungs and such. So we're trying to balance a lot of things there, and we're very excited. Thanking our donors tonight. This event is covered by generous patient donors over the years. About $75 is what's been typically given. Over 4,000 people have helped with this over the years with donating money, and that's really been an incredible thing. Over $7 million since 2006. The impact of philanthropy is manyfold, but one is its flexibility. We can buy your beer tonight with it, and we can take an exciting idea that the government says you need 2 more years to prove to us that could work, and we can tackle it now. And of course, we're training the next generation, which is one of the most wonderful parts of this job. We are our wonderful undergrads, our clinical research fellows, Aaron and Neha this year, helping all patients around the world get information without any charge. And that's what many people have wanted us to do. They said, my dad wasn't treated properly. Please make sure that other people's doctors have the information that they need to try and treat this disease well. The undergrads are incredibly, over 100 people apply for 2 spots every year for this position. So if you thought they were bright when you talked to them and met them, you're right. The philanthropy supports every team member. There's a little pie chart next to every face here. The proportion of the pie chart that's green is the proportion that's covered by philanthropic support. And you can see it's big and enabling and allows us to do crazy things like this event that don't make any financial sense, but are the right thing to do. Philanthropy and the Fred Hutch Cancer Center, we're in the middle of what's called a rebranding. The Seattle Cancer Care Alliance is merging with the old Fred Hutch Cancer Research Center to become Fred Hutch Cancer Center, FHCC, and merging then in a partnership with University of Washington. All of our cancer care, all of our philanthropy will happen basically under one umbrella, and it's going to be seamless and it's going to be great. But we're in a growth period at the moment. Any donations you make to Merkel cell carcinoma, whether it's to UW or Fred Hutch, will come to this team. And we're super lucky that in this process, although we moved from UW to Fred Hutch for our philanthropy, Christine Chan and several of our colleagues, Rebecca Allen and others, were able to move over to Fred Hutch. So we're just keeping the same partnership, and that's just been super wonderful. So I'm going to tell you 2 things about immunotherapy tonight. This is the layout of our talk tonight. And I like this little map on the bottom, that this is the patient experience: diagnosis, initial treatment, surveillance, recurrence, and recurrence treatment. Every one of those things can be, should be improved. We were in terrible shape in the past with the recurrence. Then it got much better with immunotherapies. There's still a long way to go. I'm going to start with immunotherapy updates, mostly focusing towards the end here. So as I mentioned, advanced Merkel cell carcinoma, we're talking about not the little primary thing on the arm or the face, but when it spreads to the liver and other parts of the body. That used to be a disaster. It's still very serious, but it used to be a disaster, and a revolution has happened. So what do I mean by that? Prior to 2017, in this pie chart, if you had advanced Merkel cell carcinoma, this is your chance of benefiting from chemotherapy for more than a year, 5%. Not good. solidly 60-65% were yellow, and they would have a lovely shrinkage of their cancer, but it would usually only last a few weeks. And then a chunk is in red and never benefited from chemotherapy. Today, there's a lot more green, 10 times more green. About half of patients will have persistent benefit, but we still have about half that only have temporary benefit in yellow or never had benefit in red. Usually we're talking about from one initial immune therapy drug. Our goal for the next 5 years is to increase that 50% to 75%. It'll probably take not one drug, not one pathway, but a collection of things creatively applied to patients, but I am optimistic that we'll get there. We're already seeing evidence of benefit, and we have specific plans to get there, and we'll tell you about just a few of them tonight. Last thing I will mention is a question we get all the time from our patients and from around the world. When should we stop immunotherapy after a good response? Immunotherapy is expensive and has side effects, obviously. And most of the original trials stopped the drug after 1 or 2 years. So we don't have great data about what happens after that. This is one of the things if you were on the lab tour, you see we're tracking whatever 1,600 patients, and we're getting a lot of data from them. We're trying to balance here the possibility that you need the drug in an ongoing way here on this side, and the fact that it's expensive and it's inconvenient, and it can have side effects by goosing the immune system into possible autoimmune problems. So here's what we've learned. It's not a complete story, but we're starting to think we have a pretty good idea. Your chance of having the cancer come back if you stop maybe a year after you've had a good response depends on how fully the cancer was eliminated. If it was gone based on scans, we can't see anything at all, that's called a complete response. And only 27% of those patients had their cancer come back after immunotherapy stopped. If the response was only partial, a little more than half had their cancer come back. And we know this is relevant and that they still need the drug, because when we give the drug back, the cancer shrinks again in a chunk of those cases. Not every one, but in a good chunk of them, indicating clearly the drug was useful in an ongoing way. So what we're moving to with good justification is infrequent dosing rather than a cold turkey stop. Turns out these drugs in some ways stick around in the body for not just a couple weeks, but really several months. So especially after the intensive battle is fought, we can shift to less intensive dosing. And we think that continuing some treatment for at least 1 to 2 years after the best response likely makes sense. So that's the high level on that burning question that gets asked essentially almost every day to our team. So I'd like to start now with patient story number 1.
My name's Larry Maloney. I grew up in New York City, 64 years old right now, live in Asheville, North Carolina. And the biggest sledgehammer that ever hit me in my life, because I've never really been sick, but in June of 2021, I had a pimple kind of on my forehead. And after about 2 or 3 weeks, it didn't go away. And I'm kind of a hypochondriac. And so I went to the dermatologist. I said, this doesn't look right. Luckily, he did a biopsy. He didn't turn me away. And I got the diagnosis of Merkel cell. What really helped me is that first weekend, I was just trying to find out information. I went on the internet and I was looking for different articles and things like that. And Dr. Niem's name came up a bunch of times. And I said, I'm going to try to contact Dr. Niem. So this was on a Friday. And I did, and he responded on Saturday. And I said to myself, if one of the leading doctors in the world is going to respond, that gave me a lot more hope that, we're going to be able to tackle this. When I was there, said that, they do a circulating DNA test. And I did that, and I have a high tumor mutation burden. And also they found that I have some aggressive mutations. So I think having that information is good to know. So anyway, I had everything looked great and everything is fine. 3 months later, I get a recurrence, found a couple other pimples on my head, did a PET scan, had a 1.5 centimeter tumor in my lymph node in my neck. And what was, well, I don't want to say great, but my local team here, um, conferred with Dr. Neum and his team. And what Dr. Neum did was he said I should go on Keytruda. Did the Keytruda in a couple of weeks. The tumors were gone. I got really lucky, and so far so good. There's not just hope, but there's a very good chance that with the doctor's help that you can beat this. How did the diagnosis affect me? Well, when I found out, I made a pitcher of old fashions, which gave me about 4 drinks, and that was my initial response to it. But after about 8 hours of feeling sorry for myself, I said, okay, that's done, and I need to learn as much as I can about this to see how I could beat it. The hardest thing I had to do was tell my wife and my kids about it because I felt like I was letting them down. I mean, I'm 64, I'm in very good health, and all of a sudden something like this happens. And, you say to yourself, why me? And a friend of mine who had cancer beat it so far. They said, well, why not you? Are you better than everybody else that you shouldn't have something like this? And it said, it kind of told me I need to buckle down and do whatever I can. After my 8 hours of feeling sorry for myself, I said, I am not going to let MCC define my life. I'm going to incorporate it into what I'm doing. And over the past over a year, I have not missed a beat at work. My social schedule is bigger than it ever was because I'm even leaning more forward with, recognizing how precious every day of life is. And so I am truly lucky about how things are going. For support, my family has been great about this. My wife was fantastic. If I was to give some advice to someone who's newly diagnosed, engage the experts. I was fortunate that my doctors here in Asheville, North Carolina, they've seen a couple of cases of Merkel, but not that much. And I told them about that. I'm, talking, with Seattle, and my local doctors at any point of a decision would consult and defer to what Seattle says. So to any of the people who may be remote or they may not be around or it's hard to get to a city, get your local doctors, working with the team and stay positive. And Dr. Neum said to me, I said, well, what if Keytruda doesn't work? He goes, then we're going to go to the next thing and we're going to go to the next thing. And there's multiple bullets in the gun to be able to try to figure out, how to beat this. So in the future, I mean, I'm going to keep staying on top of all of the research and the advances. If it comes back or if there's, I start hitting some obstacles, I'm going to power through it and figure out what the next thing is. I want to give a sincere thanks to all of you. You saved my life and probably going to save a lot more lives. So just keep doing it. And I am so grateful for all of you.
Thanks, Larry. I know you're here somewhere. There you are. Okay, thank you, thank you. That's an interesting comment. We're not. We're helping, but we could not do this without your help donating blood, donating money, donating tissue, allowing us to follow over time. It's absolutely a partnership. Thank you. Alex is going to talk next about blood tests. This relates to the surveillance phase here, when initial treatment indicates the cancer is gone, but we want to catch it as early as possible in that chunk of patients in whom it's going to come back. It's going to tell you about the AMERC test that we've had for a little over a decade, and the new kid on the block that addresses some of the deficiency. AMERC only works for patients that make those antibodies. And so the new circulating tumor DNA test can help also for other patients. Alex? Alex is a wonderful member of our team, headed for medical school soon, and interested in focusing on all of you, tracking hundreds of patients, making sure your tests get done at the right time, at the proper exam results get acted on the right way. Alex, go ahead.
Absolutely. Thank you so much, Dr. Niam. Hello, everyone. My name is Alex Fu, and I am a research coordinator here at the Niam Lab. And my job is to manage patient blood tests. And the importance of that is detecting disease early so that we can maximize treatment options. Now, why detect MCC recurrence early? Well, MCC has a high recurrence rate of 40%, while melanoma has one of 7%. And that means that MCC recurs often. And it also grows rapidly. When we detect MCC recurrence early, we catch it when it's controlled and localized. But when we catch it late, it progresses and it's much harder to treat. And so overall, early identification of MCC recurrence allows for more shots on goal. We can try plans A, B, and C, and say if plan A doesn't work, we have B and C to fall back on. Whereas if we detect it late, we only have, say, plan A. And I liken this to an analogy of a shot clock in basketball. When you have 24 seconds on the shot clock, you have many opportunities to pass and shoot the ball multiple times. But when you have just 8 seconds left, you have time for just one shot. And relating this back to MCC, when we detect disease early, we have many options available. Now I'll show you a timeline about how MCC surveillance has improved over the years. Before 2010, we used full skin and lymph node exams using the naked eye, as well as imaging such as CT scans. But in 2010, we introduced the idea of blood tests, and blood tests are more sensitive than scans and also more cost effective. And the first one we introduced was the AMERC blood test. Now, as Dr. Niam said, this is the test that measures the antibodies against the Merkel virus in the blood. And this test is widely used even today and very successful throughout the US and internationally. But it has one caveat. It only works for patients who produce antibodies to the Merkel virus, or patients that we call virus positive, as seen here. Now, we needed to develop a test that worked for both patients that are virus positive and virus negative. And we did this in 2021 when we introduced the circulating tumor or ctDNA test. And the reason this test works for both patients that are virus positive and negative is because all MCC patients have tumor DNA. They have MCC tumors, and thus those tumors have tumor DNA. And first, I'll talk to you a little bit more about the AMERC test before I touch on that new kid on the block that Dr. Niam was talking about. So the AMERC blood test detects antibodies against the Merkel virus. And when those antibody counts go up, it gives us an early signal of recurrence. Now, again, there are some caveats. As I touched on before, it only works for patients that produce antibodies against the virus, which is around 50% of MCC patients. And additionally, it's also less reliable after multiple recurrences because the test was made to test for the first recurrence of MCC. Now, with the circulating tumor or ctDNA test, we address some of these weaknesses. Amazingly, MCC tumors can release tumor DNA into the blood. And that's not to say that they're metastasizing or progressing through the body in any way, but they just release these fragments of DNA that we can detect. And so in the first ctDNA test, we take a portion of the tumor from a biopsy or a surgery, and we identify a patient-specific mutation from the tumor DNA. It's very unique to the tumor, and it's not found in any other cells in the body. From there, we collect the blood, and we look for that specific piece of tumor DNA, that tumor signature in the blood. And with subsequent tests, all the patient has to do is go get their blood drawn, and we can collect the blood sample and test for that specific sequence. And here's ctDNA in practice. Now, to orient you, vertically here is the ctDNA level, which is essentially the amount of tumor molecules in the blood. And over here horizontally is the time from diagnosis. So overall, I'll show you a little bit about how the levels change over time. Now, in initial treatment, the patient starts with a positive ctDNA level, which indicates that they have disease. And with successful initial treatment, that goes all the way down to 0, which we call not detectable. As we continue the surveillance phase and that stays at 0, we consider the patient to have no disease. But as soon as that increases and becomes positive, that gives us a signal that the disease is coming back and that we need to use the usefulness of imaging to look for the disease. And as you can see here, we performed a PET scan on the patient and we found a neck tumor. Now from there, since we've localized the disease, or we found the disease, we can treat that recurrent MCC. And as you can see, we were successful and the disease is now not detectable again. It is completely gone from the system. And amazingly, ctDNA as a test is also incredibly useful because the level it measures also correlates with how big the MCC tumor might be. And so this gives us a very important measure of disease burden. So for MCC tumors, even as small as half a centimeter, the size of a green pea, up until say an orange or even larger, we see an exponential increase with the size. And now you might be wondering what the red and blue dots are. The red dot is actually the virus positive patients and the blue dots are the virus negative patients. And because they trend along this exponential line, it shows that this test works for both virus positive and virus negative patients. Additionally, there's a level below 1 for ctDNA where there's actually tumors that are too small to be detectable on the scan. But incredibly, ctDNA as a test can actually detect these tumors. It gives us a positive level of ctDNA, and that indicates that there is MCC in the system, even though the scans say that they can't find anything. And so as a summary of key points, ctDNA works for over 95% of MCC patients, and we believe that number may even be higher. It works for virus positive and virus negative patients, and the only patients it doesn't work for is when the tumor is too small to sequence or if there's insufficient tissue. And additionally, both tests, AMERC and ctDNA, are incredibly useful today. They're very sensitive, and we can use them for MCC surveillance. They detect disease very early, and that gives us more shots on goal, more treatment options. And they can also avoid expensive and time-consuming scans when the tests are negative. We can save the usefulness of these scans for when the tests become positive and we know there's disease. And with that, I'll answer any questions from our Zoom chat, and thank you to my live audience. I'll talk to you after this. Thank you so much.
No questions yet? Okay, so people can ask remotely if you want to put in chat, but I see 2 questions in the room. We'll take really quick ones. Yeah.
So I'm currently doing the Natera testing. Do those results automatically feed back to you?
That's a great question. So when you do the blood draw for Natera, the kit gets sent to the Natera company, which I think is in California or wherever their headquarters are, and they'll do the resulting, and we go to their portal and we get your result from online. Yes.
Does that test carry over to any other type of cancerous tumors?
So Natera actually isn't Merkel specific, or the Signatera test is not a Merkel specific test. It actually does carry on to other cancers, yes.
Yeah, it's approved for any cancer being tracked with immunotherapy and for colon and breast and probably others in the future.
Is ctDNA test available outside of the United States?
I think technically it's available outside the United States. I should ask the question, can you get the ctDNA testing outside the United States? It's kind of complicated. Even in the United States, there's no real data published on it yet, and it's not sort of recognized yet. We think that that's going to change in the coming months. But technically it is available outside the United States. just with the bureaucratic headaches of submission and such.
Yes.
Question.
Personal question. My AMERC went down after I had radiation my second time, and then it's shot up, and my ctDNAs have been negative. And I just had another blood draw, and I won't get that result until.
So I won't go into detail about the specific situation, but the two tests are looking at different things, and the immune system can get turned on, and antibodies could, in theory, go up for one reason or another, and that may or may not directly indicate the amount of tumor. So we are actually doing heavy duty head-to-head analyses. 95% of the time they give us the exact same result, but sometimes we are learning new things by doing the comparisons, which we certainly don't know, as Alex said, which one's capable of detecting the smaller tumor, and we're still learning about that.
Thank you.
Do we anticipate FDA approval for the ctDNA test for Merkel?
I think it's technically already FDA approved for other things. Whether they specifically indicate it or not, I think we are thinking the data in the next year or so, partly based, there's 6 universities coming together to analyze data. We were just showing you our data, but there's 5 others that are gonna combine. We think that's gonna push it over and allow it to be reimbursable by insurance. Okay, and how are we on time? Who's watching time here? Okay, I don't know what that means in terms of how much we have to to go, but all right, we will maybe go ahead. Yes, there I get this official signal that we got to move it. All right, Geraldo. Okay, come on up. So Geraldo is a wonderful graduate student doing his PhD in the lab together with Justin Taylor, who's also here tonight, who's an expert on B cells, and I am not. So this is a great example of bringing people together. Geraldo is really motivated, really smart, and long time focused on B cell biology. He's going to tell you why you should care about that, but he's got a challenge. But some people think that's really funny. Okay, but one reason is the test you just heard about. The AMERC antibody test is totally dependent on B cell function. The other reason is why Justin and I and Geraldo are all excited about the concept of understanding these cells, because it might be an important key to treat the cancer more effectively. Go ahead, Geraldo.
Thanks, Paul, for the introduction. No pressure. Just, and before I even start, I like starting with a joke. Apparently the jokester of the lab, and I want to point out my hair. That was my COVID hair from 2020, which when my grandma came to visit me in 2021, she said, "You better cut that or else." Hence, I've kept it short since. But in all seriousness, before we get into B cells, I'd like to give a little bit of a background on how exactly Merkel cell carcinoma, more specifically virus-positive Merkel cell carcinoma, arises, because that's very important for me to set the stage into explaining in hopefully eloquent terms how B cells work, because they're amazing cells that are difficult to study because of their complex biology. So let's start with a normal cell in your skin. It's, for the most part, your skin is a protective barrier that prevents many infections from getting through into your body. But sometimes it itself can get infected by viruses such as the Merkel virus. And for the most part, this particular infection is benign. Most of us actually get it during our childhood or throughout adulthood. And as I mentioned, it really doesn't cause many troubles. But occasionally, in very rare circumstances, about 1 in 3, oops, I lost. Wow. Okay, it's back. Okay, so for drama, occasionally, in about 1 in 3,000 cases in the US, this viral DNA from the Merkel virus, there's an accident that happens. You can call it like a freak of nature accident that happens, where the viral DNA integrates into the DNA of the host cell. And we lost it again. Okay, I don't know. Am I moving this too much? Okay, sorry about that. But bottom line is that viral DNA fuses with the cell's DNA and it causes that cell to start producing this viral piece that shouldn't be there. It shouldn't be made by your cell. But suddenly that viral piece is actually what leads to uncontrollable cell growth. We call these oncoproteins, things that cause cells to reproduce indefinitely. And ultimately, that is what actually leads to Merkel cell carcinoma in the virus positive cases. And though, of course, there are major issues with this, it actually provides a great tool for us to investigate how your immune system is fighting off tumor cells. And the reason as to why that is because that tumor cell inherently has something that shouldn't be there, the viral protein. That viral protein, your body knows it shouldn't be there. And therefore, this is a unique way in which your immune system can be like, that cell has something wrong with it. We need to find a way to get rid of it. Now, of course, that doesn't always work, and that's why we do the research that we're doing. And essentially, we're trying to understand how your immune system recognizes the uniqueness of tumor cells, in this particular Merkel cell carcinoma. So now that I have introduced a little bit about the biology, then we can get into the B cells. And as Alex mentioned before me, the B cells are actually the ones that make antibodies. And let's say that you have your Merkel cell, and that Merkel cell has that viral piece in it. Upon cell death, by either some sort of therapy or just natural tumor death that happens because the tumors grow too big, okay. Again, maybe, am I glitching or is it the computer? Okay, just kidding. But that cell fragment, oh boy, okay. I don't know. It just flickered. I don't know.
You think it's this connector?
I think it's just it's getting bumped.
Okay, let's just not touch it.
Okay. Whew. But in any case, as I was saying, that viral piece gets released upon cell death. And now that it's outside of the cell, in your blood, oh boy. Okay. In your blood, you have an ocean of B cells that randomly surveil in and out of lymph nodes, trying to find things that do not belong to your body. And out of millions of these B cells that circulate through your body, one of them happens to actually be able to naturally identify that viral piece in Merkel cell carcinoma that doesn't belong. So once that random B cell that can naturally recognize that viral piece becomes activated upon binding to that viral piece, it becomes what we call as activated. It now knows that there's something out there that it recognizes and wants to kill it. So then that B cell eventually matures, and upon maturation, it releases a bunch of antibodies into your blood specific for that viral piece. And that is actually what we detect in the AMERC test. The product of the B cell that became activated upon recognizing that viral piece that is only present in your Merkel cells. And essentially, when we look at the AMERC test, we detect those levels to predict recurrences, because that viral piece is only present in the blood, or should in theory only be present in the blood when the Merkel tumor is back. Now, for the most part, antibodies in this particular type of cancer don't actually play a role into killing the tumor itself. And the reason as to why that is because these antibodies cannot penetrate the cells. And as I mentioned, the viral piece lives within the cell, and therefore the antibody itself that is against that viral piece cannot penetrate the cell and kill it. However, we're very excited to find some preliminary data, and many other researchers across the US have found that in different cancers, B cells might not just be antibody secreting cells. They might not just be releasing antibodies. They might actually be contributing to anti-tumor immunity. And let's say, and that happens in like the early stages of the B cell maturation. So let's say that you have a B cell that can recognize that viral piece that is exclusively Merkel cell carcinoma. That B cell then uptakes that viral piece, chops it up inside its cell, and then the B cell has this magnificent ability, or we think that has this magnificent ability to essentially speak the language that T cells can recognize, and the T cells themselves can then kill tumor cells. So On top of that, not only do B cells can speak the language that T cells recognize to kill tumors, they can also provide other signal boosters that will enhance the ability of these T cells to kill tumor cells. And though we haven't shown this in very conclusive marks, we do have some exciting data that we want to explore. I want to explore as a graduate student. And hopefully, soon enough, or if not me, then by future fellows, graduate students, and postdocs, we could potentially come up with new B cell therapies to enhance the ability of T cells to kill tumors and generate better therapies to treat recurrences as well as other aggressive forms of Merkel cell carcinoma, and maybe even potentially use what we're learning from Merkel cell carcinoma, apply it to virus native tumors, as well as any other cancer out there that we could find B cells that can kill tumors. And with that, sorry, that may have taken a little longer than expected, but I just want to highlight what Paul said, that this is the epitome of what a collaborative environment the Merkel team is. Every single part of the research that I have done exemplifies that. Collaboration between Justin Taylor, a B cell immunologist who's an expert in like identifying and engineering B cells. Paul, a Merkel cell carcinoma physician, all the clinical fellows, the graduate students. Thomas, who actually works on T cells and likes calling B cells boring all the time. But in reality, the B cells are not just boring. They're helping the trashy T cells do their job.
You have any ideas? There is something mysterious going on.
Yeah, sorry about the glitching.
Yeah, that's definitely, you weren't anywhere near the computer when it was misbehaving. Okay, great. Plan B, more direct in. Okay, thank you so much. Okay. So do we have any questions? I think maybe we'll move on, would be my thought right now, and you can submit questions later. Okay, so I'm gonna now introduce Dr. Song Park, who many of you knew when in her prior life as a clinical research fellow, but now she's on our faculty in dermatology and is leading some clinical trials, and she's going to give you an overview of these clinical trials at these various phases where they can impact the experience of patients and help the cancer not come back. Thank you, Song.
Hi everyone, my name is Song Park. I was prior one of the clinical research fellow, and I'm working as one of the physician in the MCC team and leading several clinical trials and research projects as well. I am very excited to update you with the new and ongoing clinical trials that we've been working hard. And please note that those trials that I introduced today are highlighted trials that we are mostly involved in Seattle, and I will share further resources to learn more about other clinical trials at the end of my talk. With that, let me start with a vaccine trial that we are very excited to open in Seattle. So someone may ask, why are you using vaccines to prevent the MCC from the recurrence? So you've heard this many times. MCC has 40% high risk of the recurrence overall. And the actual risk of the recurrence varies from 10 to 90% in an individual patient, depending on the each risk factors. And then currently there's no FDA approved therapy after the initial treatment of MCC with the surgery, radiation, etc., other than just watching it closely. So we need options for patients to lower their risk of their disease from recurring from the first time. So how about using a vaccine? So MCC vaccine could fight the MCC being caused in this specific trial caused by the Merkel virus. So how does MCC vaccine could work then? So after the initial treatment having the MCC, the immune response targeting the MCC tend to fall over time. And this is amount of the MCC immunity, and then this is time from the surgery and radiation and the treatment. So this may contribute the high risk of the recurrence over time. So our goal of using the vaccine is that by giving the vaccine to the patient, we could boost the anti-Merkel virus immunity over time. So the MCC targeted T cells now, the immune cells, can eliminate microtumors hiding your blood and hiding somewhere in your body. So we are very excited to open the first ever human trials for MCC vaccine in Seattle. And I'd like to appreciate my mentors and colleagues, and especially Vivian and Sam, who helped a lot to open this trial in Seattle. So let me go briefly about how we operate this trial in terms of logistics in Seattle. So this is a 6 months of the trial. They start with a baseline scan to make sure there's no clinically detectable disease. And then we give vaccine 4 times to the patient 1 month apart. This will be delivered with a needle-free injector that looks like this. The patient have less pain, and then we can deliver the drug more effectively to the body. And at the end of the 6 months, we will have a final scan to make sure that everything looks good. And each visit, we'll draw a small amount of blood to check the patient is doing well and then to check the immune response after the vaccination. This will work best for patients with a virus positive MCC who had no evidence of disease for more than 1 year but still has some recurrence risk in their body. Our goal of this trial is really to test that this vaccine is safe to give. And then how does Merkel virus vaccine boost the immunity and how much does it boost? So we are looking into those parameters, looking into the blood and other clinical parameters. So with that, let me go over to other trials using immune therapy for higher risk patients for time. So, well, we are working with the vaccine to lower the risk of the recurrence. But can we prevent MCC recurrences by immune therapy? So I'd like to go over 2 trials that we are having in Seattle, led by Dr. Bhatia, and Wenny here is doing a critical role with leading the ATOM trials, operating the ATOM trial here in Seattle. So those 2 trials use immune therapy. ATOM trials uses avelumab, and STEMP trial uses Keytruda. And ATOM trials are for patient more advanced disease involving the lymph node at the time of the diagnosis. And the patient will receive immune therapy or placebo, which is the saline that looks like immune therapy, but actually not the drug. And it will be very closely observed for 2 years if there will be any recurrence event. And the STEMP trial, the patient could have local or lymph node involvement at the time of the diagnosis, and the patient will receive immune therapy or just be observed over 1 year. Both of the trial, the patient should have no evidence of disease on the study entrance. And then we are excited to run this trial over time and look into the data. So this was about how we want to lower the risk of the recurrence or prevent Merkel from coming back. But how about when the immune therapy does not work anymore? We need something else to fight the cancer. So I will go over 4 different clinical trials. 2 are active in Seattle. The other 2 are upcoming in Seattle. So ATTACK trial led by Dr. Beach in Seattle, we use T cells that are reprogrammed to recognize the Merkel virus, then infused back into the patient. And the patient will continue the current approved immune therapy like avelumab or Keytruda and other treatment. Execute trial led by Dr. Bhatia here uses the immune stimulating drug that is directly injected into the tumor, and then patient will continue currently approved immune therapy. And 2 upcoming trials, I will go over the POCA trials that were briefly mentioned in other previous slides, is using the expanded T cells that respond to the Merkel virus. And ATR trials is a drug that kills rapidly dividing cells that causes the immune system to attack the cancer more effectively. So with that, all of those trials that I went over are available in Seattle, and several other trials available across the nations. So you're welcome to look into the available options for you. And as I work on these clinical trials, I realized that those trials not just something that we offer for you. We really work with you to make the progress in the field and learn more about the disease and the treatment. So I look forward to partnering with you all by learning more about the trial or even participating in the trial if it works for you. So I mentioned that there are more resources that you can learn more about the clinical trials, MCC clinical trials. The clinicaltrials.gov are more like a comprehensive list of all the trials available in the nation. It's not best patient friendly. So the clinicaltrials.gov made a research match, which is more patient-friendly search engines that you can use. So you can use the QR code to go directly to those websites, and then you are welcome to work on. And with that, I'm happy to take questions. Yes. Yeah, good question. So yeah, got it. So why the vaccine trials only for the virus positive patients? So with a vaccine, well, we now have a lot of vaccines. You can imagine what we are really targeting, and the sort of low hanging fruit for us is that the virus positive tumors kind of shares this similar targets all across like the tumors, so we can use it. Correct me if I understand anything wrong.
We have two more questions. How many patients will you enroll in a trial, vaccine trial, and how long will you follow these patients?
Good. Yeah. How many patients we are enrolling on the trial? We are aiming 8 patients for now for the phase 1 trial, which is to check those immune responses and safety. And we are looking forward to opening the further trials. For now, we are recruiting 8 patients. And then how long we are following up. In the trial, per the protocol, we are only following up the patient for 6 months. However, MCC surveillance, as you saw in the previous slides, it's many, many years of the follow-up. So yeah, we do the standard of care after that.
Okay, awesome. Okay. Let's see here. Ready for story number 2, and thank you so much for doing the switch here so we can show that.
Good evening. My name is Sammy Cravens. I'm from Oklahoma City. So as far as Merkel cell, I'll start my journey with in 2009, I was diagnosed with lymphoma and successfully beat that cancer. Was under quite a long treatment plan, as is pretty common with lymphoma. And then in 2010, while I was still undergoing chemo for lymphoma, I was diagnosed with renal cell carcinoma of my right kidney and lost a portion of the kidney, not the whole entire kidney. 2 totally unrelated cancers, totally just surprise. And in 2013, was leaving on a trip to celebrate with some of my girlfriends, just getting over all that and all their involvement and participation and all the meals and just kind of a thank you trip to everyone. And so I went on my trip and came back and I noticed that little bump was getting larger, but it was still very, very small. So I went to the dermatologist and she's very effective and very efficient. And she said, you know, I'm just going to take a sample of this. We'll do a quick biopsy. I'll call you with the results. I don't think it's anything, but it's not like anything I've ever seen before. So the next week I was on my way to work and I was actually pulling in my parking place and she called me and she said, hey, Sammy, are you driving? And I said, oh, actually, I'm just in the parking lot pulling in my office at work. And she said, okay, well, get parked. I want to talk to you about the results of your test. And I said, okay. So keeping in mind, I'd had cancer twice before. I'm thinking, yeah, this is not good. So I pulled in my parking place and I said, yeah, what's up? And she said, well, listen, the results of that biopsy came back and it's a very rare form of skin cancer called Merkel cell. And I said, oh, okay. I said, so what do you need to do? And she said, well, I think we need to have it completely removed, and I'm going to send you to a surgeon downtown and he'll do the work. And I said, well, does he usually do this sort of work? And she said, I don't really think anybody around here does this sort of work. It's very rare. And sure enough, I ended up with Dr. Neum and his team. I was there, I think I talked to him that very same day. And that night he called me. And then right after that, I figured out a time to get there. And as everyone knows, Tuesdays are clinic days. So I think I was there the next Tuesday and met with Dr. Neum and the team and felt so relieved because I was talking to somebody that knew what they were doing. And so we started the journey. I ended up coming back to Seattle for surgery and got that taken care of. And we developed a treatment plan. And all in all, it was a journey and we got through it. And I felt really great about the relationships that we had built and what needed to happen. So that was in 2013. In 2016, I noticed in one of the scans that there was something going on with that partial right kidney that I had. So they took a look at that, did a biopsy of that, and determined that Merkel cell had come back to that kidney. And so that was right at 3 years. So they completely removed that kidney. Just about 3 or 4 short months, it came back again in another area internally, and it was metastatic at that point. Did single fraction radiation and then started on Keytruda. And for 4 years, I did Keytruda. So that was through from 2017 through 2021. So the medication was halted in 2021 and took me a little bit of time to kind of get my body back to normal. But the entire time I've worked, I've really never been off more than just the requirement for the scans or a surgery or whatever it might be. So I'm back at work, of course, as I have been the entire time, and I feel great. I feel better than I have in years. So I think that one of the things that helped me the most was just the positive attitude. I was able to maintain that because I'm surrounded by really positive people. My family's really positive. My friends are really positive. I had lots of comfort. It was important to me that I wanted to say thank you to all them. And I think my attitude, the positivity that I was able to maintain throughout the time frame really helped with that. And people wanted to come over and hang out and, bring food and come by my office and bring lunch. And the work that I do is really meaningful to me because we have a lot of people that we have a large company now, and we have a lot of people that count on me to be here and to just be a ray of sunshine in the office. So I try to do that for people, and people were giving back to me in that respect as well. I've also been working with Integra's Cancer Institute, which is one of the medical providers here, and they have a lot of activities for people going through cancer. And they have a mindful meditation class, they have a yoga nidra class, and I'm now in my second round of meditation classes. And so I've learned a lot about that and learned how to just really kind of control my emotions and my feelings. And that's been very, very helpful. So one of the things I found really helpful from the time I spent in Seattle was learning about a support group that the University of Washington group and Hutch has. It's called Merkel Cell at Google Groups, I believe. And so that's a group run by a man named George. And there is so much information and people are so positive in that group. You don't just hear the negative, and people really latch onto one another and are so encouraging and happy to help and provide advice and not medical advice, but just like, hey, you might check with this group to help you with travel expenses, or this is a great place to go in your area. We've had a lot of people in the group that have found resources in this area or that area. So I have found that to be really comforting. And now that I am through the thick of it and can really supply some positive stories. I'm happy to always respond to someone that asks a question. So that group has been a really lifesaver for me. And I appreciate all the people in that group, as well as George, the man that runs the group. So that's sort of my story. I'm excited to feel great again. I know that, we never know what's going to happen tomorrow, and I'm just really thankful for every single day.
My name is Judy, and I'm from Bainbridge Island, Washington. On February 16th, I was in the shower and noticed a growth the size of half an avocado between my left armpit and breast. It was not there on February 15th, and I was scared. After advanced ultrasound followed by a needle biopsy on February 25th, I received the diagnosis of Merkel cell carcinoma. Merkel cell carcinoma is a rare and aggressive form of skin cancer. The diagnosis left me feeling shock, disbelief, and in utter terror. I have cried and sat with my husband and ran through all the dark thoughts possible about my future. The odds of me getting this cancer did not work as well as when I bought a lottery ticket, but yet here we are. Speaking of here we are, how lucky, yes, I said lucky, we are to be in Seattle, where the foremost medical expert in this field are working right now, ready to tackle this battle with us. I may not have drawn the winning lottery ticket, but I sure as hell won the lottery of Merkel cell carcinoma experts who are working to cure this beast. My tumor team of Dr. Neem, Dr. Bird, Dr. Colahan, Dr. Basha, and their fellows, nurses, radiologists, PAs, all work together to form my treatment protocol. This gave me a feeling of comfort, knowing they were working and meeting weekly to discuss my case. I also knew that I could access them through my chart with questions and concerns and would always receive a timely response. After a 7.5-hour surgery and 25 radiation treatments, and currently being in a blind clinical drug trial, I know I am in good hands. I found the support of family, friends, and Seattle Cancer Care Alliance team to be the ticket to my positive attitude. Thank you all for giving me hope and the will to kick cancer's ass.
I think we flipped it around this year. And for the folks online, thank you very much for joining. And if you didn't make it, or you know somebody who didn't make it and wants to, we will be having little segments of this on the web so that you can watch if you missed something or something. It's time for dinner. The folks who are on the extended Merkel team, if you can come down, we're going to take a group photo, which we haven't done in 3 years, which will be pretty cool. And thank you all for coming and contributing in so many different ways. As I was thinking back, I'm thinking of the people who are running the AMERC assay at University of Washington, and the folks who are looking under the most sophisticated microscopes to assess why people do and don't respond, and the many of you that have donated blood and money and tumor and I just couldn't, I think, Shalender, we couldn't have dreamt of where this would be today, huh? So many people to thank. Thank you all. Live Oak, great job. And have a great evening. We'll be out there in a few minutes. You guys get started with the food. Oh, and there's going to be a little more music too. So, okay.